So we have the thyroid gland and then multiple follicles in there. And there's a lot of follicular cells. And all the cells in the one follicles, they work in the synchronized manner. So if the cells are making the thyroid gland, all the cells in the follicles make the thyroid hormones. Okay? And then once the thyroid gland gets the signal, the follicular cells now they're releasing the thyroid, the hormones. Then all the cells in the same follicle gonna release the thyroid hormones, the same manners. So that's thyroid hormone synthesis and the secretion, and then we have to understand how that happens. And then at the end, you need to know what is the function of the thyroid hormones. I already give you some idea. Measure the regulators of the metabolisms in our body. Okay, and then of course we need to go through the pathological condition. Again, hyperthyroidism, hypothyroidism. Those are the very important pathological conditions you need to went through. So this is it, but I think it's very important. All right. So the anatomy of the thyroid gland, again, the thyroid gland has the two lobes. It's connected by the isthmus is in front of larynx. So we have, maybe you can feel the thyroid gland in this area. Okay. So this is the largest endocrine gland. Weight is about the 15 to 20 grams. And then this is highly, the blood perfused organs. So it's about the 5 ml per grams of tissues per minute. The blood go in through the thyroid arteries, and it's going to go out through the thyroid vein, right? So that is really, really highly perfused organs. So we discussed about the liver, highly perfused organs. So what is the most abundant type of the cells in the liver? That was in the questions, right? In the exams. What was it? So most of you guys picked the hepatocyte. But the key is the liver is the highly vascularized organs. So if you're looking at the number, endothelial cell is way more than the hepatocyte because of a lot of blood vessels. Okay, so the kidney, highly perfused organ. Liver is a highly perfused organ. Thyroid gland, highly perfused endocrine gland. What is the common thing? All of these three, this is important in the metabolism. All right. So the metabolic product being produced, metabolic waste being produced, this has to be go through the circulations, right? So this one is a highly perfused endocrine gland. And then there is some adrenergic and then cholinergic innervations. That means the function of the thyroid gland is heavily influenced by a neurological regulations, right? So that's the kind of things in the anatomical structure. And the histology. So we have the thyroid gland. If you're looking at the histology, the slides in here, we can see the various components in here. So the first one is the follicles. So we have the follicles like this. There are numerous follicles in the thyroid gland. Okay. So the lobes of thyroid gland is basically the aggregate of the spherical the follicles like this, lots. And then this follicle is composed of the follicular cells, and then each follicular cell has to be connected to each other. It's very, very tight. So very tight junction is existing between the cells, which means we have the follicles, and there's something being produced, and they store the inside of these follicles. Because between the cells, we have the very tight junctions. Inside of the follicle content is not going to be leaked out. It has to go through a cells, right? So the thyroid gland, follicular cells, is the cells are making the thyroid hormones, and it's going to be in charge of releasing the hormones at the end. Okay. And then the next one is the colloid. So we have the gel-like structure and the component in here inside of the follicle that is called the colloid. Okay. So this is the colloid again, the thick gel-like substances in the lumen of the follicle. This one is primarily composed of the thyroglobulin. The thyroglobulin is the globulin that can store the thyroid hormones. So we're going to talk about it more. So the follicular cells making the thyroglobulin, and the thyroid hormone actually attached to the thyroglobulin and being stored in this inside of the follicle in the forms of the colloids. Okay, and then the follicular cell. This is the cells that makes the T3 and T4. So we have thyroid hormones here. The thyroxine is the T4. T3 is the 3-iodothyronine. So among these two, T4 and T3, you must know this. Which one is the biologically active, the forms of the thyroid hormone? T3. T4 basically have the 4-iodine attached. T3, 3-iodine attached. So that's the difference. So the T4 in the blood circulation can undergo the deiodination. That means T4 can become T3, the biologically active forms of the thyroid hormones. So anyway, these the T3 and T4 will be produced by a follicular cells. And then these the follicular cells, we have the cells. The basal membrane is very close to the capillary networks. So once the cells make the hormones, it's going to be stored inside of the follicle in the part of the colloid. And then when the cells get the signal, these the thyroid hormones going to get into the cells again. And then these from the thyroglobulin and T3 and T4 is going to be released into the capillary, and these capillaries are going to go to the systemic circulation. So that the blood vasculature, the blood vessels, and the cell is located very close to each other. So it's very efficient, the delivery of the hormones to the circulations. Okay. And then the cells in here is the parafollicular cells. We have the follicles, and the cells next to the follicle is called the parafollicular cell. It's going to make the calcitonin. Have you ever seen this hormone before? The calcitonin is the opposite to the PTH, and then PTH is parathyroid hormone. Calcitonin is the thyroid hormones. I hope you still remember that one I mentioned in the Chem 1. Okay. So regulations of thyroid gland, again, we have the circulations. The circulatory thyroid hormone is going to go down, and it's going to stimulating the hypothalamus, and the hypothalamus will making a TRH, and then inhibiting the inhibitory factors, right? So TRH is the thyrotropin releasing hormones, and then the inhibitory factor is the somatostatin. So the somatostatin goes down, the releasing factor is going to go up, then it's going to stimulating the anterior pituitary gland, thyrotropes. Again, anterior pituitary gland has a lot of trope cells, and then among that, the thyrotropes is going to make the TSH, which is very important hormone, TSH. You should be familiar with that. And this TSH, the thyroid stimulating hormone, is going to stimulate the thyroid follicles now, and then these the thyroid follicles will release the thyroid hormones. Then the thyroid hormone level is going to go up, and then increase the thyroid hormone is going to provide a negative feedback signal to the hypothalamus and the thyrotropes and the thyroid gland, so that the thyroid hormone release is going to be slowed down. So this is one of the examples of the negative feedback mechanisms. So in our bodies, again, we have the decrease of thyroid hormones. It's going to stimulating the hypothalamus, and the hypothalamus is stimulating the pituitary gland, and the pituitary gland will stimulate the thyroid gland. It's going to make the TSH, the thyroid hormones, right, T3 and T4. Once the T3, T4 is being released, it's going to do functions like this, increase the metabolism. Again, thyroid hormone is the key regulators of the metabolic rate in our body. And then it's going to increase the growth and the development. So you can say that if you, a baby, don't have enough thyroid hormones, the growth and development can be problematic, right? And also the increase the catecholamine effect. I want you to pay attention to this. So increase the metabolism. Energy production is going to go up. And those energies being required to increase a sympathetic nervous systems, right? So again, the thyroid hormones can increase the catecholamine effect also. Okay, so this is kind of summary of the biological function of thyroid, the systems, but we're gonna revisit later on the more. So this is the thyroid hormone synthesis pathway. Okay, so again, this is the thyroglobulin, the globulin proteins, and you can see a lot of Y here, the Y, alphabet Y, which means the tyrosines, right? The tyrosine is the source of this the thyroglobulin. So we have the two different the structures in here. One is the thyroxine, which is a T4. So you can see 4 iodines being attached. And then we have the 3-iodothyronine, which is T3. You can see 1, 2, 3 iodines being attached. Okay, so we must know the kind of the numberings on these, the ring structure. So you can have carbon number 1, 2, 3, 4, 5, 6. Okay, so you can say that number 3, number 5 is the iodinated. This is important. So we have the first rings is a 1, 2, 3, 4, 5, 6 is a counterclock, the Y's. And we have the second ring, so through the O linkage in here, that's the prime. So we have the prime, 1 prime, 2 prime, 3 prime, 5 prime, or something like that. So 1, 2, 3, 4, 5, the primes. Yeah, 1, 5, right? Something like that. So if you're looking at the numbers in here, in the T3s, we have iodine, the missings in the 5 primes, right? You have to know the difference between T4 and T3 in terms of iodine location. Why this one is important? So later, as you're gonna see, the reverse T3s are T3. So that's the other, the T3s, but the iodide missing in the 5 position instead of the 5 prime positions. And these, the reverse T3 is functional as the regulators. So if you have too much of the RT3s, actually the thyroid hormones released from the thyroid gland can be slowed down. Those kind of things. So we're going to talk about it more later on. So this, probably the rest of the times today, I'm going to use this one. And I think I'm going to be very happy if I can finish this one. So we're going to go through that, the thyroid hormone synthesis pathway. Okay. So the first, we need to make a thyroglobulin. The thyroglobulin contains a lot of, tyrosines, in that the thyroglobulins. Okay. So this, the thyroglobulin, the synthesis happens in, endoplasmic reticulums. And then it's going to be packaged, the thyroglobulin was being produced by the endoplasmic reticulum. It's going to be packaged in the Golgi apparatus, and then it's going to move toward apical membranes. So you have to use your imaginations. We have the cells, base membrane, apical membranes. Basal membranes, we have connections or the close to the capillaries, right? So once the endoplasmic reticulums make the thyroglobulins, have a lot of tyrosine in there, and then it's going to be packaged in the Golgi apparatus, and then those packaged thyroglobulins are going to be moved toward apical membranes. Apical membrane is going to face a lumen of the follicle, right? So that happens, and then in the follicular cells, the cell has to concentrate the iodide from the circulations. Okay. So if you compare the iodide concentrations in the circulations and the inside of the cells, iodide concentration is about 200 times more than the circulations. So actually the follicular cell has already a lot of iodide, 200 times more than the circulation levels of iodide. Iodide, we have to rely on to the dietary source, right? So that we have to move the iodide from the low concentration to the high concentrations. So you must have iodide transporter, which is ATPase, right, the required ATP to move something against concentration gradient. Okay. So again, the iodide is coming in, and the follicular cells can use those iodide on there. Okay. The thyroglobulin, the assemblies, so iodinations of the thyroglobulin can happen. We need the thyroid peroxidase. So in the tyrosines, thyroid peroxidase is going to do oxidizing it, and then the iodide can bind you to it. Okay. So once we have the tyrosine residue in the thyroglobulin, tyrosines, and these peroxides are oxidizing it, so the iodide is attached, right? Can you follow this? Tyrosine, one iodide attached, that's called monoiodotyrosine, MIT. Tyrosine, one iodide attached is the MIT. And then it is possible tyrosine, two iodide attached, right? It's called the DIT, diiodotyrosines. So in the globulin, the thyroglobulin, a lot of tyrosine residues, and now iodination happens, so we're going to have the MIT and DIT. Okay, and then after that, MIT, DIT formed, and then we're going to have another couplings. These couplings also require a thyroid peroxidase. Okay, so if you have DIT, MIT binding together, it's kind of 2 plus 1, 3, right? And then DIT, 2 plus 2, T4. So we're going to have, in the thyroglobulin, we're going to have the various forms of iodinated products like this, right? So we have the tyrosine residues, so likely the red indicating the tyrosines, okay? And then if you have the T4, the 4 iodinations happens, then this is the T4. And also we can have the 3 iodinations, probably this is a 5' position, so the T3. And also we can only have the DIT and MIT also can bind into it. So this MIT and DIT can dimerize again by the peroxidase. And then also we can have the reverse, the 3 iodotyronides. Again, instead of the 5' position, we're going to have the 5 positions that deiodination happens, right? And then is the reverse 3 iodothyronine, the RT3s. One pathological condition is associated with that is the euthyroid sick syndromes. So these syndromes has the kind of abnormal T3 and T4 the level in the blood. It's because it's abnormal thyroid feedback systems, Okay. So the thyroid gland is functioning, but we have too much of these RT3s, so that the TSH level is not much stimulated. So if you somehow increase RT3, which is not functioning, but it's gonna slow down the thyroid hormone release from the thyroid gland. As a result, it's gonna cause the euthyroid sick syndromes. Again, knowing the deiodination place is kind of important on that. Again, so we're looking at the thyroid hormone synthesis, and then we're going to go on to how the thyroid hormones can be released. So it's going to starting from the pinocytosis in the apical membrane of follicular cells. Again, follicular cells make the thyroid hormones, and the thyroid hormone, thyroglobulin is released into the follicle. And once the cells get the signals, so now the pinocytosis happens. It could be macro, it could be micro. So the colloid, the part, is going to be internalized inside of the cells. And then this colloid droplet is migrating toward the basal membranes of the follicular cells, where it's going to be close to the capillary, the networks. And then on the way to that, the basal membranes, the colloid droplet is going to be fused with the lysosomes, which has a lot of the proteolytic enzyme in there. So these lysosomes going to hydrolyze the thyroglobulin to its constitute and the amino acids. So we have in the thyroglobulin, we have T3, T4, mid, and DITs. All of these can be detached by these the lysosomes. And T3 and T4s and other iodinated amino acids can be released into the cytosol now, right, because of that the lysosome. And then it's going to go to the circulation, and some other amino acid can be recycled, and even some of the iodide can be recycled for the next cytoglobulin, the synthesis of that, those kind of thing. So let me use this, summarizing it. Okay, so we have this. Yeah, I'm gonna use this. So this is the thyroglobulin. I mean the thyroid, the cells, right? So we have the basal membrane, blood, and then the apical membranes facing the lumen, right? So in the basals area, we have iodide transporter, which is the ATPase, so that the iodide can be moved from the vasculature to the cells, right? And we have a lot of iodide inside the cells. So we have a lot of iodides available inside the cells now. And then the endoplasmic reticulums, cytoglobulin, the synthesis is happening. And then this one will be packaged in the Golgi apparatus. So these to package the cytoglobulin precursors going to be moved toward apical membranes and waiting in there, right? We have the cytoglobulin precursors, okay, a lot of tyrosine in there. And then again, the iodide from the vasculature can be moved into the cells against the concentration gradient through the iodide transporters. The iodide transporters are located in the basal membranes. This is an ATPase. Now, this iodide can move toward apical membranes where the cytoglobulin is located already. And then we have the peroxidase. So these peroxidase do oxidize the tyrosine residue, and then the iodide can bind it to it. So because of that, is the peroxidase, we have DIT can be formed, and then another coupling, the DIT can happen, so it can have the T3 and T4 can be formed. So at the end, we have the cytoglobulin carry DIT, T3, T4, all of them. Can you follow? So this is the thyroid hormone synthesis pathway happening in the follicular cells. And then these, the cytoglobulin, can be stored in the lumen of the follicle as part of the globule. So again, follicular cells make one follicles, and all the cells in the making the follicles are going to function in the synchronized manner. If the one cell make the thyroid hormones, all the cells in that the same follicle making hormones, and then dumping the hormone into the lumen of this follicle. Okay? Now, your cells get the signals, let's say the TSH being released, and then stimulating these cells. And what happens is, these the thyroid hormones will be get into the cells through the micropinocytosis or macropinocytosis. So this, the colloid droplet, is going to be moving toward a basal membranes. And then inside of the cells, we have the lysosomes. And these, the colloid droplet and the lysosomes are going to be fused. And then the lysosomes have a lot of enzymes that can do the proteolysis. So the cytoglobulin carry T3, T4. And these guys will be detached from the cytoglobulin because of these, the lysosome enzymes. Okay. And then these T3, T4 can get out, right, from the cells, and then go to circulation. And then this and this iodine can be deiodinated, and it can be recycled. And then the amino acid in here can go endoplasmic reticulum and can be used to make a cytoglobulin. Okay. So this slide is going to explain the step-by-steps how that the thyroid hormones can be produced and how that the thyroid hormones can be released from the follicles of the thyroid gland. Again, so again, between the follicular cells we have the very tight junctions. So the the content in the lumen of the follicle cannot travel through the tight junctions. So it has to go through the follicular cells, and the follicular cell really kind of very accurately regulated the release of thyroid hormones. Any questions about this? Okay, so yeah. Okay, I'm gonna stop in here, and I really want to finish the endocrinology, the in-persons. So next week I'm gonna do endocrinology again in the in-persons in here, and in the meantime, the toxicology I'm gonna put it in the campus as the recorded versions. So again, toxicology and the therapeutic drug monitoring. So maybe I'm gonna post both the lectures, even though we discussed the therapeutic drug monitoring in the Chem 1. There's a huge connections between those two, so I'm gonna just post that one in the campus. Okay. All right, I think we can finish next week. Right, and then hopefully I can save some time next week. I don't know if it's possible, but I'm gonna try, and then I'm gonna go over briefly to go over something we discussed in the Chem 1 and the Chem 2. Okay, probably 30 minutes should be good enough. 30 minutes cover Chem 1 and Chem 2. Maybe it's impossible, but I'm gonna try. Okay, so what are we supposed to do? So this afternoon, actually, I want to tumor marker. Actually, that's the one, right, for the lab materials. So go through the tumor marker, and the next week, again, is going to be the practicum. So if you have any questions about the practicum, please shoot me an email. Okay, right. I'm going to stop in here. I'm going to let you guys go.